Okay. Hi, good evening. My name is Nandika. I'm a PhD student at IIC Bangalore, and welcome to the problem solving session for experimental biotechnology. Today, we're going to discuss content related to week 9, which was dealing with cell biology, microscopy, and flow cytometry. Of course, if you have any doubts, please feel free to ask me, add them in the chat, or whatever. And in the meantime, I'll discuss some concepts and problems related to what we learned this week. So, in this week, what we've been discussing is mostly the concept of microscopy, or how we can observe um, the cells and other biomolecules and so on. And just to give you an idea of what scale looks like in biology, yeah, this is what we can see with our naked eye, right? If we just hold up our finger, this is what we can see. And so this is on a scale of about 20 millimeters. And maybe if you look really, really close, you can see all the ridges in your skin, right? You can see like the uneven surface of your finger, right? And that this is on a scale of about 2 millimeters. And this is really the limit of what we can see with our naked eye. If we were to go down by a power of 10 again, okay, to a scale of about 0.2 millimeters, we would then start seeing the actual structural features of the tissue itself. We would be able to see the cells, the connective tissue, and so on, right? And this is something that we can no longer see with our naked eye. And again, if you keep going and magnifying this image further and further, so you look at this little square over here and magnify it 10 times, then you can see all of the individual cells with the organelles inside the cell, right? And this is on a scale of micrometers, right? So the human eukaryotic cell is usually between 10 to 20 micrometers in diameter. So this is the kind of magnification that we're going to need if you want to look at cells and want to study what's going on in them. If instead you actually want to look at the organelles themselves, then again you'll have to magnify this 10 times. And then you can see the mitochondrion, you can see the outer membrane, inner membrane, so on, all the ribosomes over here. Again, if you want to look at the proteins themselves, then you have to magnify another 10 times. And then you can see the ribosomes, the proteins, and then if you keep magnifying, eventually you will get to the atomic structure, right? So this is on a range of nanometers now, 2 nanometers. And this is basically the maximum resolution that we can achieve using the instruments that we have today, okay, where we can look at the density of the individual atoms themselves. Okay, and we'll discuss how we can go from here to here today. So as we discussed, there is a huge range of sizes of biological specimens, right, ranging from things like yeast and bacteria all the way into individual animal cells, organelles, ribosome, proteins, etc. And because this is such a large range, we need different ways of studying them. And the most common microscope that you probably will have used in your lab is a light microscope. Okay? And that can take us to a range of around 500 to 100 nanometers. Okay, so it can cover a range of this where we can see things like bacteria, yeast, cells, and so on. Okay? We may not be able to see organelles, but that's okay. We can use other methods for that. But for now, we'll just discuss what a light microscope does. So, a light microscope basically exploits the properties of a lens. So, you may be familiar with something called the lens formula, where this is a lens, okay, a thin glass lens. And a lens, by property of a lens is that it has something called a focal point, okay. And what the meaning of a focal point is if I have light rays, parallel light rays which are coming from infinity, then the focal point is the point at which the lens will focus these parallel light rays. Okay? But what if I don't have light rays which are coming from infinity? What if I have an object here? Okay, it is beyond the focal point, but I have some object over here. What happens is that the rays from this object pass through the lens and form this magnified image over here on the other side of the lens. Okay? And if we define this object distance between the lens and the object as p, and the distance between the lens and the image is q, then we can determine what the magnification of this image is. So you can see that this image is actually bigger than the object itself, right? So that means that by passing the light through the lens, we've obtained a magnified image of the object. And through this relation, we can determine what the magnification of the image itself is. And by using many combinations of these lenses, we can actually obtain a highly magnified view of our object. So just a simple light microscope would look something like this. I mean, I know I called it simple and it doesn't look very simple in this photo, but there's only 3 major lenses which are there in this system. Okay, so in this light microscope, you would keep your sample over here. Okay, specimen holder, you can see it's called over here. So you would keep our sample over here. Obviously, we need light to fall on our specimen, right? That's how we're going to see it. It's called a light microscope. So we need a light source. So the light source is coming from a lamp which is down here. Okay, so this lamp is providing illumination, right? It's going to provide light which is going to fall on a specimen. Between the lamp and our specimen, there's something called a condenser. Okay? So the condenser collects and focuses a cone of light that illuminates the tissue on the stage. Okay? So this is a series of lenses that basically collects all the light which is coming from this lamp and brings it onto our sample. Now here above the sample, you will see something called the objective. The objective lens enlarge and project the illuminated image of the object towards the eyepiece. Okay? So this objective is going to be what actually provides the magnification of our image. So you can maybe see that there's one objective over here and there's one more objective lens over here. So there are different objective lenses which give us different magnifications of our object. And then you can see that the light which is collected by the objective is now being passed through something called the eyepiece, right? This is how we're going to observe. We're going to observe through the eyepiece. And in the eyepiece itself, there is another lens. Okay. So the 2 eyepieces or oculars magnify this image another 10x and project it to the viewer. So here we are magnifying the image, and here we're going to magnify the image again. So this whole setup is going to be how we obtain a magnified image of our object. There are basically 2 kinds of light microscopes that are usually used. So this is the setup that we just discussed, where the light is coming from below, our sample is kept over here, the objective is here, and our eyepiece is here. So this is called an upright microscope. Here you can see that the distance between the objective and the sample is very small, right? So we can easily keep a slide over here, but if you want to keep a flask, okay, or something bigger, then it will be very difficult for us to fit it in this space. Okay? And if you do want to observe a sample like that, then we would use something like this, which is called an inverted microscope. Now, as the name inverted implies, instead of having the light source at the bottom, now the light source is on top. Okay? This is the light source. You have the condenser here, and the objective is actually under our sample. Okay, so you can see the objective lens kind of sticking out over here. And this whole space is available for us to keep our sample. What is going to happen is that our objective lens will allow us to focus on the bottom layer of the sample, which is here. Okay, so light is going to come from on top. It's going to be collected by the condenser. It's going to be focused on our sample, going to be magnified by this objective lens, and sent to the eyepiece over here. Right? So just in a diagram fashion, this is what an upright microscope looks like. As we discussed, lamp, condenser, object, objective, and eyepiece. Here you'll see something called stage focus control. Okay, these are the same knobs that we hear and here. Okay, and this is basically allowing you to adjust the height of the sample so that it comes, so that the image that you form is in focus. Okay. And similarly, the inverted microscope, as we discussed, the light source is on top. Then you have the condenser, you have the stage, objective, and the eyepiece. So as we discussed, the objective lens is what is going to provide the actual magnification, or like the majority of the magnification of our sample. Here you can see this is something called a turret, a lens turret, and you can see there are multiple objective lenses. Here, and you can see here it's written 5x. This one is written 10x. And all of these others will have different numbers written. So this is the magnification that is provided by this lens. So this is going to magnify your image 5 times. This is going to magnify 10 times, and so on. Okay. There's also a lot of other information that's also written on this objective. But that is something that you have to worry about maybe at a later stage. So the total magnification that we're going to get from a microscope is going to be, we're getting some magnification from the objective, but like we said earlier, we are also going to get some magnification from the eyepiece. The eyepiece is going to provide a magnification of 10x. So the total magnification of the microscope is going to be the product of these two. Okay. So for example, if we have an objective which is 60x, but the final image that we see is not going to be magnified 60 times because it's also passing through the eyepiece, which is providing an extra 10x. So the overall magnification of this will be 16 to 10 or 600x. Okay. So this you have to keep in mind when you're reporting the magnification of anything you look at through a light microscope. So normally in a light microscope you will have objectives of 4x, 10x, 20x, 40x, and 100x. So 100x is usually the highest objective that you can use in a light microscope. And so you will get 100x magnification from the objective and 10x from the eyepiece. So total magnification will be 1000x. Okay. So that will allow you to see things like cells and maybe some organelles within the cell. So like we discussed, there are many things which are written on the objective. So the most important thing that you have to keep in mind is the magnification. And here they will give you another number which is called the numerical aperture. Okay. The numerical aperture is basically telling you about the light collecting ability of the lens. Okay, we'll discuss in the next few slides what numerical aperture actually means. But it determines the resolution obtained and the amount of light gathered. The higher the numerical aperture, the better the resolution that you're going to get from your lens. Next to numerical aperture, sometimes you will also write something else like oil. Okay, and this is something called the immersion medium. And we'll discuss in the next few slides what an immersion medium is and why you would use it. Okay. If nothing is written over here, that means you have to use it in air itself. Here they'll also mention the cover slip thickness for which this lens is made. And here is something called the lens type. So this is a slightly technical piece of information which is telling you about some of the corrections that are present within the lens. you'll get names like achromat, fluorite, apochromat, plan apochromat, and so on. And these are telling you some of the slight corrections that are there within the lens. Okay. But basically, all you really need to remember is that the best lenses are called plan apochromat lenses, in which they have corrected for variations in color and spherical aberration. Okay, so this is what you will find in high-end research microscopes. So, as we just discussed, one concept which is very important when talking about objectives is something called numerical aperture. So, numerical aperture is basically given by this formula, nu, which is the refractive index of this medium. Okay, basically a light is passing through this medium and then is getting collected by the objective. Okay, so this is the cone of light that is collected by the objective, and the half angle, this half angle within the cone is called alpha. And you can see if the cone that is collected by the objective is bigger, that means the objective is able to collect more light, then alpha is also more, right? This angle will be more correspondingly. So this is a lower numerical aperture objective, and this is a higher numerical aperture objective. So the numerical aperture is called nu, which is the refractive index of this medium. For air, nu is 1 multiplied by sine alpha, which is this angle. And obviously you can see if as we increase this angle, the numerical aperture of the objective is going to increase, right? So this has Na of 0.45, this is Na of 0.75. But you can see obviously that there is one more way to increase the numerical aperture, which is to increase nu, right? Which is to increase the refractive index of the medium through which the light is traveling. So when we do that, that is what is called an immersion medium. So here you can see light is passing through a sample. It's going to pass through a cover slip, and then normally it would pass through air, refractive index of 1, and then it's going to be collected by our objective lens. However, there is beyond a certain angle, okay, so like light rays traveling at this angle are not going to be collected by this objective lens. However, if we now change this medium, if we basically what we're going to do is keep our sample, put a drop of oil, and then bring it to our objective lens. Okay, we're going to touch the objective lens to the oil, so there's no air left over here. So now light is going to travel from our sample through the oil into our lens. And now you can see the same angle of light ray which was not getting collected on this side is now collected by the oil and taken into the objective, right? That means a greater amount of light is collected when we change the immersion medium from air to oil. And so what we see correspondingly is a increase in the numerical aperture, because remember this is the formula for numerical aperture, and now we've just increased nu. So 1.4 is generally the highest numerical aperture that we can achieve in a light microscope. Okay. Another concept we have to understand in microscopy, all microscopy, not just light microscopy, is the concept of resolution, which is what is the smallest distance between two objects that we can distinguish using our microscope, right? So just in a crude sense, when we look at things with a light microscope, if there are two different cells, right, we can easily tell that these two cells are separate objects. Even within the cell, okay, I can tell that this nucleus and this mitochondrion are two different organelles. But what I can't tell is the individual position of each ribosome within the mitochondrion, right? spots, I don't have the magnification to tell the difference or to tell the exact position of these dots within the mitochondrion. So these are below the resolution limit of our microscope. So there is a definition of what is a resolution limit, and it is given by this. So essentially, when we image any point object in a light microscope, it will form a pattern like this. Okay, this is just a product of a diffraction. Okay, so you will get this diffraction pattern for any point object. And the minimum separation at which we can make out these two point objects separately is called the limit of resolution. Okay, this is something called the Rayleigh criterion. So the Rayleigh criterion states that this distance is r. The distance from the center of this bright spot to the middle of this dark spot. This much is r. And our objects should be separated by this minimum of this distance r so that we can separate them visually. So like these two spots we can tell are two different spots. But when they've come slightly closer, okay, now these two bright spots have started to merge. And now we can no longer tell whether this is actually two spots or a single spot. So we say that these two spots are not resolved. Whereas these two spots are at the limit of resolution, okay, because they're separated by the Rayleigh criterion. Yes, this is called the Rayleigh criterion R, which is equal to the radius of this disk. Okay. So resolution, or R, is given by this formula: 1.22 lambda by 2 times the numerical aperture. Okay. Obviously, if we just take these constants, the formula will become 0.61 lambda by the numerical aperture. So here you will see one factor that we haven't discussed so far, which is lambda. So this is the wavelength of the light that we are using to illuminate the sample. So that means depending on whether we are using a red light or a blue light, the limit of resolution is actually going to change. Okay? So the lowest wavelength light that we can probably use for light microscopy is blue light. Okay? So if we look at, so red light will have lambda of around 600 nanometers. Let's say we take some red light, we say lambda is 600 nanometers. Okay. For this R, we'll say Na is 1.4. Okay, we'll use a good oil immersion lens. For this, for red light, the limit of resolution will be the same thing, 0.61 into lambda, that's 0.61 into 600, divided by the numerical aperture, which will be 261 nanometers. Okay. So this will be the limit of resolution for red light. Instead, if we use blue light, okay, with the same lens, our limit of resolution will be 213 nanometers. Okay. So basically, the limit of resolution, which is defined by this criterion for a light microscope, is around 200 nanometers. Okay. So we cannot see objects which are separated by a distance of less than 200 nanometers. So if you remember back to our initial slide where we talked about the different scales of objects that we have, we cannot observe individual proteins using a light microscope, right? Because they're separated by a distance of less than 200 nanometers, right? So this is a basic limitation because of the nature of light that we are using. Now you may say, what if I use something with an even smaller lambda, right? And then we get into something called electron microscopy, which we'll discuss later. But just for visible light microscopy, we do get into this resolution limit of around 200 nanometers. Another thing in light microscopy is because of this resolution limit, we actually cannot see objects which are smaller than the diffraction limit. And so, even if the object is like this, okay, what we will actually end up seeing in the light microscope is this. Okay? So, any objects which are below something called the diffraction limit will give us spots at of the same size as the diffraction limit. Okay? So, objects which are this big and objects which are this big will end up giving us the same spot. This is just a limitation because of the nature of the light that we are using. Okay, so now that we've understood the basics of how light microscopy works, we can discuss a couple of variations that are there in light microscopy. So, one thing that we obviously want in light microscopy is we want to be able to see the differences between various parts of our image, right? So, I mean, different, if we're looking at a cell, then different organelles within the cell should appear different, right? Otherwise, we won't be able to make out anything. So, that means we need to have some contrast in the image, right? Maybe some parts should be bright, some parts should be dark, and so on. Now, unfortunately, if we just image a cell through bright field, just simple light microscopy, there isn't a lot of contrast within the cell. Okay, if you just image a cell straight up, you may see the boundary of the cell. Maybe you'll see some dark region, some granules, and that's about it. Okay. You will not be able to make out any features of the cell because most of the cell is made up of water. Okay. So there isn't a lot of contrast within the cell naturally. So that means that we have to find ways of enhancing the contrast within the cell. So some ways are you can stain part of the cell, right? So artificially you're adding color or contrast within the cell. Okay, so this is going to make different parts of the cell appear as different colors. You might have done something like hematoxylin and eosin staining and things like that. So, or even if you've done staining with things like safranin, right? So you are specifically staining some parts of the cell so they appear different in color. Another option is something called dark field microscopy. Okay, so in normal bright field microscopy, what was happening is that all the light was going to come like this. It's going to come straight, and most of the light is going to enter the objective. So dark field microscopy is the opposite. The light is coming at an angle. Okay, so if this light is not interacting with the sample, it is not going to enter the objective. So that means the background where nothing is present, that is going to be black because there's going to be no light from that region. In order for the light to enter the objective, it has to interact with the sample and it has to scatter, right? So if this is scattered sufficiently, it will enter the objective. So say this is interacting like this will enter the objective, but this won't. And as a result, you're basically only collecting the scattered rays. So this is one way of just increasing the contrast within your cell. This is something called dark field microscopy. Another thing that we can use is make use of the fact that different parts of the cell have very slight differences in refractive index. Okay, that means that some parts of it may be thicker, some parts of it may be denser. So you can see that like this part of the cell is thicker compared to this part, right? And obviously the refractive index of the cytoplasm, it's mostly water, is going to be different from the refractive index of air. So when a light wave is passing through this region of a different refractive index, it's going to have a slight phase shift compared to any light which is not passing through the cell. Okay. What we can do is basically amplify these differences in phase between the light passing through the sample and the light which is not passing through a sample. Okay, so if we have a difference in phase and then we allow these beams to interact with each other, what is going to happen is going to get either constructive interference or destructive interference or something like that. which is going to basically increase or decrease the brightness of our image. Right? So we are making use of the differences in the phase to generate contrast in our image. So this is something called phase contrast microscopy. Okay, so it's called phase contrast, or is slightly different method called differential interference contrast microscopy. Okay? So these two methods don't require staining, we're just making use of the natural features of the cell to increase contrast. So just an example of why we need to do things like this. So this is an image you would get from a bright field microscope, right? And you can kind of see that, okay, maybe my cell boundary is like this, but I can't really see any features of the cell, right? So like we discussed, if we do use phase contrast microscopy, like we're causing the differences in phase, we're using those differences in phase, and we're causing the light waves to interfere with each other, which is why we're getting either an increase or decrease in the brightness of our light. Now suddenly we can see the features of the cell much better, right? So here, I mean, I don't think here you can even tell that the cells are present properly, right? You can see maybe the outline of some nucleus, maybe the outline of a cell. But if I look at it in phase contrast microscopy, suddenly it's far more obvious where the cells are, where the nuclei are, right? You can even see some organelles inside the cell. Right? So this is phase contrast microscopy, this is differential interference microscopy. So these are different ways of looking at unstained cells, just changing the optics of the microscope to increase the contrast within the image. So just an overview of all the different light microscopy methods that we can use. First off, the simplest method is bright field with an unstained specimen. Here light passes directly through the specimen. The image has low contrast. Here you can see in this image, you can barely make out the cell boundary and maybe the nucleus within the cell. The first way of generating contrast is to stain it with various dyes. Right? So here you can see that now the cell has become red and the nucleus has become black. Right? And now we can easily make out the difference between these two regions. If we don't want to stain the sample, we can use something called phase contrast, where we're using variations in density within the sample. And now suddenly we can make out the features much better. We can use optical modification. So this is a different kind of phase contrast. Okay. Or what we can do is start labeling different parts of the cell. And the most common and most sensitive way to do this is through something called fluorescence microscopy. Okay. Fluorescence microscopy, the locations of specific molecules in the cell can be revealed by labeling the molecules with fluorescent dyes or antibodies. So here, what we're going to do is label specific sections of the cell using different fluorophores, and then image that together to get an idea of the different regions within the cell. And these are all just variations in fluorescence microscopy. Okay, so we'll discuss a little bit about confocal microscopy. These are more advanced methods, but these are all very powerful techniques that we can use using essentially fluorescence labeled antibodies and specific labeling of cells samples. So before that, we can just quickly go through some questions related to light microscopy. So which type of microscope uses visible light to illuminate the specimen? Okay, so this is a light microscope. The answer to this is actually, it's not an electron microscope, not an atomic force microscope. The answer is actually fluorescence microscope. Yeah. What is the function of the condenser in a microscope? So like we discussed, the condenser is the lens which is present. If this is my lamp, my condenser is the lens which is present over here, which is just here below the sample, which is responsible for collecting the light from the lamp and bringing it to the sample. Right? So, what is the function of the condenser lens? It is to control the amount of light entering the objective lens. Right? It is not going to have anything to do with the focus or magnification of the specimen. It is just there to control the illumination. What is the maximum magnification achievable with the light microscope? So, like we discussed, the highest objective that we can use with a microscope is 100x. The eyepiece is providing additional 10x. So, the highest magnification that we can achieve is 1000x, which is the product of these two. What is the purpose of staining specimens in microscopy? The purpose of staining specimens is to increase contrast and visibility, because normal bright field microscopy does not have a lot of contrast. What is the role of the objective lens? So the objective lens is the lens which is present above the sample. Okay. So what it's going to do is collect the light from the sample, okay, magnify it, and then bring it to the eyepiece. Okay. So the role of the objective lens is to provide the initial magnification that we get for our sample. Okay. What would be the maximum resolution that can be achieved if a sample is illuminated by green light and observed through an oil immersion objective with numerical aperture of 1.5? So the maximum resolution would be 514 divided by 2Na. Okay, which would be 514 divided by 3, which is 171.3. Okay, so we will go with 171 nanometers. What is the purpose of using a phase plate in phase contrast microscopy? So as we discussed, phase contrast microscopy is making use of the natural differences in density and refractive index within the sample to increase contrast. So the phase plate is used to increase contrast of transparent specimens. Okay. So we've discussed basics of light microscopy, and we discussed that there's a technique called fluorescence microscopy that we can make use of to label specific sections of the cell of our interest. So we'll take some time to discuss what is fluorescence microscopy and how does it work. So this is the basic phenomenon of fluorescence. Yeah. So this is something called a Jablonski diagram. It's a common representation of this phenomenon. And these are different energy levels within an atom. Yeah. So these are different energy levels within an atom. So this is the ground energy level per se, and this is the first excited energy level. So when an electron absorbs light of the correct wavelength or correct energy, which gives it this, which is equal to the energy gap between these two levels, what happens is that it gets excited, okay, because it received this additional energy. It is able to jump from this ground state to the first excited state. Okay. Once it is in the first excited state, it is now going to lose energy. Okay, it's going to lose energy through a process called internal conversion, and then it is going to jump back to the ground state. And obviously in this process it's going to lose energy, and this energy is going to be emitted from here in the form of light. Okay. So this emitted light is called fluorescence. Yeah. Now as you can see, here we have absorbed this much light, so it's gone from this ground vibrational level in S0 to this higher vibrational state in S1. But within the S1 state, it is the electron has now dropped several energy levels. Okay, it's lost this through energy through internal conversion. So this is probably lost in the form of heat, and then it has lost this much energy as fluorescence, right? So there is some energy which is lost prior to the emission of this photon. So this photon, the emitted photon, will obviously have less energy than the photon that was used for excitation, right? So as a result, the emitted photon will actually have a longer wavelength compared to the absorbed photon, right? Because there's a loss in energy, the wavelength will increase. So if we look at the excitation spectrum, right, excitation wavelength, which is blue, for example, then the emission wavelength will always have a longer wavelength and it will have less energy. So this is what the emission photons will look like. So if I excite it with blue light, then the fluorophore will emit green light. And this difference is called something called the Stokes shift. Right? This change in towards a longer wavelength is called the Stokes shift. Okay? And this is true for every fluorophore. So this particular spectrum corresponds to a green fluorophore, but you will have similar spectra for fluorophores of all colors. Here you can see that it's not like there's one specific wavelength of light that is needed for excitation. Okay, there's basically a range of wavelengths that can be used to excite the molecule. This will be the most efficient wavelength. Okay, this will be most efficient in exciting the fluorophore molecule. And if I excite with this wavelength, then I will get the maximum emission of fluorescence, basically. But even if I use some wavelength here, okay, which is not exactly the ideal wavelength, it's a little further away, but it's still within the excitation spectrum. So it still has energy sufficient to excite electrons within the fluorophore, then I will still get emission. It's just that the amount of the emission will be less compared to this. Okay. So even if we use suboptimal excitation wavelength, we will still have the same emission spectrum, but we will get diminished intensity. Okay. So here the point being that the emission spectrum will remain the same, or rather the nature of the emission spectrum will remain the same regardless of which excitation wavelength you use. It's just that we will see differences in intensity. So this is what a simple fluorescence microscope will look like. It's called an epifluorescence microscope. Epifluorescence microscope. So again, we're considering a green fluorophore where we will use blue light to excite it and it will emit green light. So first we have to shine blue light on the sample. So this is our source. Okay, we can use a laser or maybe a xenon lamp or tungsten lamp or some light source. Okay. So here we are going to allow only blue light to fall on our sample. It's going to be reflected through this mirror. Okay, this mirror is important. It's what is called a dichroic mirror. And what a dichroic mirror does, that it is able to reflect some wavelengths and allow some wavelengths to pass through. And this is important because we are going to use blue light to excite a sample, but we are going to collect the green light which is emitted from a sample. So the green light which is emitted from a sample is allowed to pass through the dichroic mirror and into the eyepiece. Right? So that means we are not going to see any of this light that we used to excite the sample because all of that is going to be reflected. So any signal that we are going to get here is as a result of the fluorescence emission from our fluorophore. Okay. And this makes fluorescence microscopy a very powerful technique, a very sensitive technique. Okay. Here you will see a filter, and this is basically regulating the nature of the light which we are going to use to excite our object. So if we have a light source which has multiple wavelengths, then here we can have multiple filters which allow different wavelengths of light to pass through. And in this way, we can use multiple fluorophores. Right? Say I have a fluorophore which I need to excite with green light instead. Then I can change this first barrier filter, which will now it will allow the green light to pass through, and it will block the blue and the red light. Right? So now I can use the green light to excite my fluorophore instead. As we discussed, in the middle of a fluorescence microscope, we have something called a dichroic mirror. And dichroic mirrors are designed to reflect specific wavelengths. Right? So it's reflecting specific wavelengths and transmitting other wavelengths of light. Okay? So this is what a dichroic mirror would look like. It is going to selectively reflect the blue light. So this is what is going to go towards the sample. But it is going to allow red and green light to pass through. And this is just this dichroic mirror. We can have dichroic mirrors that have different cutoffs, okay, for different ranges. So what this would look like in a spectrum is something like this. Okay, so here you see 100% reflectance. This is 100% transmission. Here you can see in the blue range, okay, this is blue-green range, all of this light is getting reflected. Okay. Suddenly at around 580, let's say 550, 560 nanometers, right, 560 nanometers, you can see suddenly all the light is going to start getting transmitted through the mirror. Okay. So shorter wavelengths are reflected, longer wavelengths pass through the mirror. Okay. And depending on the fluorophore, we can choose a dichroic mirror which is suitable for, you can use a dichroic mirror which is suitable for its excitation and emission wavelengths. There's a whole range of fluorophores that we can use in fluorescence microscopy. As you can see, it spans the whole spectrum. So of course the excitation will have lower wavelength than the emission. So here's an example of some of the fluorophores that we talked about earlier. They'll have excitation in the blue range, emission in the green range. But we can also have fluorophores which are red, fluorophores which are blue, fluorophores which are yellow, and so on. So there's a whole range of fluorophores that we can use for different applications. Now, what we have is a fluorophore, but how can we use that to stain our cell of interest? So what we can do is actually conjugate the fluorophore to an antibody. And we've discussed quite extensively how antibodies are specific to certain targets within the cell. So if we have an antibody against a protein of our interest and we conjugate a fluorophore to it, and we use that, treat the cell with this complex, then this will go and bind to our protein of interest. And because the fluorophore is stuck on it, when we give the correct excitation light, we will be able to see the exact position of our protein of interest, right? Because this fluorophore is gone and stuck only on our protein of interest. Right? And this is essentially how most fluorescence microscopy works. We can use it in fixed cells. We can use it in live cells as well. Okay. So same deal, we have fluorescently labeled antibody. We've expressed it within our cell, and it has gone and labeled our protein of interest. Okay. Like we discussed in techniques such as ELISA, we can do this immunolabeling either in a direct or indirect manner. In direct manner, the antibody is already conjugated with our fluorophore. Okay, so we will use only one antibody. So it is going to bind to our protein of interest, and it has the fluorescent label already on it. As opposed to this, you can have indirect immunolabeling, where we have our antibody, the primary antibody which we have generated against a protein of interest, and we can have a secondary antibody against our primary antibody. And the secondary antibody is going to be what is going to carry the fluorescent label. So obviously this is going to, what this means is that we can use the same secondary antibody for multiple primary antibodies. So if we have a primary antibody which is generated in same mice, then all antibodies which are generated in mice, we can use the same secondary antibody, which will be an anti-mouse antibody, to detect them, and the secondary antibody will carry the fluorescent label. If you want to go for direct immunolabeling, we can actually add the fluorophore to the antibody itself, where we have reactive fluorophore. So this will react with certain groups in your antibody. So it can react with lysines or cysteines, or maybe certain tags in your antibody. So if we incubate the antibody with a fluorescent dye and pass it through a G25 column, okay, this is essentially a size exclusion column. What is going to happen is that a fluorescently labeled antibody is going to be larger than the free dye, so it is going to pass out first, and so we can purify our directly labeled antibody. Right? So we can use this for direct immunolabeling. And a direct immunofluorescence protocol will look something like this. And this is just a general protocol. You don't have to do it like this, but just to give you an idea, first we have to fix the cells. So what fixing means is that we want to make sure that everything which is in the cell remains the same way that it was in the biological state. Okay? We don't want like the positions of organelles and so on within the cell to change due to the staining procedure. What we want to capture is an accurate representation of what the cell looked like in its biological state. Okay? So for that, we need to fix the cell. So what that will normally do is involve things like cross-linking proteins and so on. So that makes sure that everything which is in the cell stays in the same place throughout our imaging and labeling protocols. So we can use something called like 1% formaldehyde. There are other fixing agents. After that, we get rid of the fixing solution. Then we will use something called blocking, because we don't want our antibody to bind non-specifically to proteins which are not of our interest. So for that, we can use a blocking buffer. This will contain something like BSA. Then we'll incubate with our primary antibody, which is what is going to bind to our protein of interest. We can wash to get rid of excess antibody, incubate with a secondary antibody, wash. Then we will use something called an anti-fade agent. Okay? This is basically to preserve the fluorescence during our imaging protocol. Okay? We'll mount the slide and we'll view it. Like we mentioned, we need to do fixation of the cells. So common fixatives, especially for fluorescence microscopy, are a combination of acetone and methanol. So if we fix an acetone, we get good fluorescence intensity, but methanol is best for preserving the biological structure of the cell. So normally we'll do maybe a 50-50 mixture of these two, and that will be probably the best fixative for immunolabeling. So this is good at preserving the immunogenic sites within our cell to allow the antibody to bind to it. When we incubate our slide with our antibodies, we want to use a moist chamber. Okay, we don't want the antibody to dry out and not get absorbed within our samples. So here we can keep a cover slips, okay, or the slide which has our sample. Add the antibody on top. Here at the bottom we'll have like moist tissue paper, and we'll close it. So this is just a moist chamber which allows us to incubate our sample with our antibodies. And as we discussed, something called anti-fade agents. Okay. So this is something that is going to allow a sample to retain its fluorescence intensity throughout our um, imaging protocol. So in the protocol, we saw something called vector shield, which is a commercially available preparation. We can also use something called fluoroguard. So these are things that you might commonly see in your fluorescence protocols. But if you're looking for agents itself, we can use something like ascorbic acid. Okay, even common chemicals like ascorbic acid can be useful as anti-fade agents. Now, depending on which fluorophores we want to use, we also have to look at which laser we're using for fluorescence microscopy. So, most common lasers will be things like argon ion or helium neon. Okay, so depending on the nature of the laser, that will also uh, determine which fluorophores you can visualize with your microscope. Yes, so for example, argon ion will give you these emission lines. Right? So your fluorophore should have emission spectrum within this range for you to be able to see it using this microscope. If instead I want to look at a red fluorophore, then I need to have an additional laser which will allow me to excite the red fluorophore. Right? So I need basically this helium neon laser in addition to the argon ion laser. So then I can look at multiple fluorophores. Another thing we need to be wary of in fluorescence microscopy is something called autofluorescence, which is a property that some of the material within the cell itself may be fluorescent um, as a natural property. Okay? So things like aromatic amino acid residues. So residues like tryptophan especially are fluorescent. So tryptophan has fluorescence where you can excite at around 290 nanometers and collect emission at 350 nanometer. Okay, this doesn't usually interfere with confocal microscopy, but if you want to visualize other things in this range, then you have to be wary of aromatic amino acid residues. So, other things like pyridine nucleotides, flavins, chitin, chlorophyll, lipofuxin, okay, these are all naturally occurring compounds within the cell that have intrinsic fluorescence. And we need to be wary of these when we're thinking about which fluorophores we want to use. um, Fluorophores don't have to be conjugated to antibodies. There are other kinds of fluorophores that can be specific to various organelles within the cell. So here, all these molecules that you can see here are actually membrane dyes. Okay, so these are all small hydrophobic molecules which are fluorescent and actually get are attracted to the membrane and label the membrane itself. Okay, so you can use this to label the cell membrane. Like membrane probes, you can also have dyes which are specific for different organelles. So these are things called mitochondrial probes. So they stain mitochondria with different degrees of specificity. So these make use of different properties of the organelle to make it to selectively label them. So these can depend on things like active membrane potential or certain molecular properties of the mitochondria. Most commonly, you'll see something called mitotracker used to label mitochondria. Similarly, we can have probes specific for the Golgi apparatus, okay, probes specific for the endoplasmic reticulum, probes specific for the lysosome. Okay, so lysosomal probes, you may be looking at things like low pH. Right? So depending on, you can have probes which exploit specific properties of your this organelle and thus allow you to label them. As we discussed, these are membrane markers, which are hydrophobic fluorescent molecules, and they can be used to highlight cellular membranes. So far, we've discussed something called epifluorescence microscopy, where we're going to illuminate the whole sample and collect fluorescence from the whole sample. More commonly, if you want nicer or more high resolution images, we will use something called confocal microscopy. Okay, and what that does is basically illuminate this region of the it's going to illuminate this region of the sample. Okay, so it's illuminated with a focused point of light from a pinhole. Lasers pass through this pinhole, and it's going to illuminate this focused point of light. And as a result, even when we collect the emitted light, we're only going to collect light which is emitted from that point. Okay, we're going to make use of another pinhole here, which is only going to collect light from this point. And any light from the background is going to be mostly discarded from the detector. So we are going to very specifically get signal only from a certain point in the sample, and as a result, we're going to get a very sharp image. And just we can combine various properties to get a bigger picture view of our sample. So we have a sample, okay, and if we image it over a period of time, okay, we can get a time lapse. So for example, you can look at things like cell division. We're just imaging the same sample for over a long period of time. So we can get a time lapse. We can take a sample, but we can image it in various wavelengths, right? We can label different parts of the sample with different fluorophores and image them separately. And as a result, we're going to get an idea of what the different regions of our sample are doing in our sample. And so we can get a multi-wavelength idea of multiple sample components within the same time. The other option is we focus on a certain XY region, but then we can take a series of snapshots of the sample at various Z heights. Okay, and that is going to basically form a reconstruction of the sample, okay, within this volume. Okay? So as we change the focus, we can move through different Z heights, and that will reconstruct the sample as a 3D volume. So all of these give us a slightly better understanding of the sample as opposed to just a single wavelength snapshot of our sample. And just quickly, we can discuss some questions related to this. So fixation of a biological sample can be done by treating it with methanol. Okay, all these reagents are used for other steps of the staining protocol. Permeabilization of fixed RBCs on a slide can be done by treating with 1% Triton X. What is the purpose of this step? So the purpose of treating with 1% Triton X is something called permeabilization. And what this does is basically it makes holes in the membrane. And because of the holes, our antibody and dye can enter the cell membrane. Okay, our antibody is allowed to enter the cell membrane. It can bind to structures within the cell. Okay, so this is a very important step if you're going to be labeling with things like antibodies. Otherwise, the antibody won't be able to enter the cell at all. How many dichroic mirrors are present in an epifluorescence microscope? So as we discussed, in an epifluorescence microscope, there's one dichroic mirror, which is going to reflect the excitation light, and allow the emission light to pass. So there is one dichroic mirror. Philippine staining can be used to study phagocytosis. So Philippine stains the cell membrane. So this is a fluorescent probe. Okay, so we have just spent time discussing light microscopy in various modalities. And as we saw, light microscopy has an inherent diffraction limit or resolution limit of around 200 nanometers. If you want to go beyond this resolution limit, essentially what we need to do is decrease the lambda of the incident radiation that we are using. And so when we go beyond light, what we start looking at is our electrons. So basically using electrons to collect information about our sample. Okay, so we'll discuss now a process called electron microscopy. There are some similarities between an electron microscope and a light microscope. Okay, so we have an illumination source, we'll call it. So we have a light source here. So we have a light source here and we'll have an electron source here. Same way, we have a condenser lens here, we have a condenser aperture here, which is again responsible for controlling the illumination which is falling on our sample. Right? Specimen is kept over here. And we have an objective lens, right, in a light microscope. In similar way, we have a series of objective lenses, okay, objective and projector lenses in the electron microscope, which are responsible for the magnification aspect. Okay. And at the bottom here, we have a detector. So this is usually going to be a screen or an electron detector. Whereas in a light microscope, we would be using an eye, our own eye, or we would be using a camera to detect the image. So here you can see like the overall structure is fairly similar. What has changed is the nature of the illumination that we're using. So here obviously we're using an electron source, which means that electrons are going to fall on the specimen and interact with it. And when an electron falls on a specimen, it can interact with it in a number of ways. Okay, we'll consider incident electrons are coming from here. And this is an atom within our specimen. Here you can see the electron can either not interact with this atom at all. Okay, this will be a transmitted electron. Here you can see the electron is interacting with the atom in the sense that it is basically curving around the nucleus. Yeah? So this is something called elastic scattering, right? Where there's no loss of energy, but it's just changed the electron path. Okay? And depending on the nature of how far this has moved from its original path, you can also get something called backscattered electrons, which are basically moving back in the opposite direction. You can also have something called inelastic scattering, okay, where energy is not conserved. That can occur when our incident electron hits an electron which is already present within the atom. And then we can get some loss of energy, which can either be through characteristic X-rays, or this electron can be actually ejected from the atom. This is called a secondary electron. Okay. So just looking at it in a different way, this is a specimen and our electron beam is coming like this. Then we'll get the electrons which pass through the specimen. We can have inelastically scattered electrons, elastically scattered electrons, and some unscattered electrons. So these are electrons which are going to pass through our sample. But we're also going to have electrons which are going to come on the same side. So these will be things like backscattered electrons and secondary electrons. So these electrons, which are basically emitted from the surface of the sample, are going to be used in something called scanning electron microscopy, right? Whereas electrons which are able to pass through the sample are going to be used for transmission electron microscopy. So these are two different modes of electron microscopy which are useful for two different methods. Okay, there are two different electron microscopy methods which can be used to study different properties of the sample. So just to recap, transmission electron microscopy makes use of transmitted electrons, which means electron beam is passing through the sample and the image that we get is a 2D projection of the sample. Okay, so we're going to be able to see the interior of the sample. Where scanning electron microscopy, the electron beam is going to interact with the surface and we are going to detect electrons which are scattered or ejected from the surface of the electron, from the surface of the sample, sorry. And as a result, the image that we're going to take will describe the surface of our sample. Obviously, since these are two different imaging modes, the setup of the microscope will change slightly. So this is a typical transmission electron microscope, which is the same as the electron microscope we discussed earlier. Electron source, condenser lens. Okay, we have the sample, followed by the objective lens, and then this detector. However, in a scanning electron microscope, you'll see the condenser lenses and the objective lens are both present above the sample. So the electron beam is instead brought to a point, okay, and this point is going to basically scan the sample like this. It's going to move in an XY direction. It's going to move like this across our sample to cover the whole surface. And across its full scan, what we're going to detect are things like backscattered electrons and secondary electrons. and those will give us information about the nature of the sample surface. So just to discuss the differences between SEM and TEM, SEM is looking at scattered scanning electrons, while TEM is looking at transmitted electrons. The accelerating voltage. Okay, high tension is basically telling us the voltage through which the electrons are accelerating. So it's giving us an idea of the energy of the electrons required. So SEM requires relatively lower energy, so 1 to 30 kV, whereas TM can take place like 60, 100, 200, 300 kV accelerating voltages. In SEM, we're going to look at the surface of the sample, so it doesn't matter really how thick the sample is. However, electrons actually have fairly poor penetrating power. So if you want to collect transmitted electrons, then a sample cannot be very thick. So you have to use a fairly thin sample. Something like less than 150 nanometers is required for transmission electron microscopy. As we mentioned, scanning electron microscopy will provide a 3D image of the surface. TEM will provide a 2D projection image of the inner structure. SEM is going to provide a magnification of about 2 million, whereas TEM can provide more than 50 million x magnification. In the SEM, image formation is because electrons are captured and counted by detectors, whereas TEM can be imaged directly on a fluorescent screen or again with a CCD. Sample preparation for SEM is relatively straightforward, whereas for TEM, you can imagine we have to make a sample that can withstand this kind of condition. It has to be a thin sample, but it has to be able to withstand these highly accelerated electrons. So the sample preparation for TEM can be somewhat laborious. And just as an example of what we can see, this will be a scanning electron microscopy image of bacteria. Right? The color is false colored, but you can see here, we can see individual bacterial cells, right, on a matrix. Whereas if we were to look at a bacterium in transmission electron microscopy, here you can see the ultrastructure of the bacterium, right? You can see the two membranes, you can see a nucleoid, you can see all these granular particles within the cytoplasm. And you can see obviously that this image is far more magnified while compared to this. So depending on what you want to see in your sample, if I wanted to see some feature of the bacterial surface, so maybe I'm looking at nanotubes, or I want to see things like flagella, then I would be more interested in an SEM image. But if I want to look at organelles within the cell, then I would be looking for a TEM image. Now, what will sample preparation look like for this? So for scanning electron microscopy, the sample preparation will start with fixation. Okay, so this will be things like dehydration of the sample, because water can interfere with the electrons. So ideally we want to get rid of the water from the specimen. So we need to dehydrate the sample, or we can fix the sample by freezing it. Okay. Then what is essential in SEM is that for most samples, we will coat the sample. We coat the sample with something to provide high contrast. And then we can use it for scanning electron microscopy. So coating is something called sputtering. So by sputtering, basically what we're going to do is between two electrodes, we are going to bombard a metal target with ions. This is going to slowly erode the metal target, which is going to deposit metal ions on our substrate. Okay? So we're going to get a thin layer of metal on the surface of our sample. So here you see this ant, which has now been covered in a metal. Usually for scanning electron microscopy, people will use gold. Okay, mostly to give a high contrast. And then when we image it using the electron microscope, we can see an image like this. So so the amount of detail we can get in this is amazing, right? You can see like the individual components of the compound eye. So this is an example of how we can harness the power of scanning electron microscopy. On the other hand, if you want to do transmission electron microscopy, like we mentioned, we need very thin samples. So we need to fix the sample, dehydrate it, And then we need to somehow make, if say we have a cell or a tissue sample, we need to cut very thin sections of the tissue sample. So for that, usually we embed it in a resin, okay, which is going to give rigidity to the sample, and then we're going to section it so that the thickness of the sample is appropriate for transmission electron microscopy. Then we can use, again, if you want to increase contrast within the sample, remember in light microscopy we were using dyes for staining. For electron microscopy, we need to use things which appear different to an electron. So we have something called electron dense materials, okay, which are going to reflect more electrons or scatter more electrons rather. So things, heavy metals like uranium and lead are usually used for staining. So maybe you can soak the background in uranyl acetate. So that way we get a dark background and our the sample will be light. Okay? So we can use staining to increase contrast, and then our sample is ready for TEM. Fixatives commonly used in electron microscopy are usually glutaraldehyde or osmium tetroxide. So these will cross-link the molecules and make sure that they stay rigid throughout the imaging process. This is what a sample preparation step might look like. Say I want to look at this tissue. Then first I have to fix it to make sure that all its components stay in place. So this is where we would use the glutaraldehyde osmium tetroxide. Then we'll go through a dehydration series. So here essentially we are replacing the water content, okay, and so how this works, usually you will use an increasing series of alcohol. Okay, so you can put your sample in maybe 50%, 60%, 70%, 80%, up to 100% alcohol. That way all the water will be removed from the sample. Then we have to encase it within our resin. Okay, so here at the end we have our sample embedded within our resin. And then we can use this instrument, it's called a microtome. Okay, our block will be held over here, and there's a very thin blade, very thin, very sharp blade, which is going to come and cut our sample. It's going to basically form, so here you can see our specimen, which is embedded in wax and resin. The microtome arm is going to move like this, and our blade is going to be fixed here. So as the microtome moves, it's going to cut very thin sections of this block. So we're going to get a ribbon of these very thin sections. Okay, so we can collect these sections and use them as a sample. For transmission electron microscopy, we can't keep a specimen on a slide. Instead, we have to use this metal grid. Okay, so usually a copper grid, which can be covered in carbon, and this is going to be the support for our specimen. So here we see the thin ribbons of our specimen. You can see how thin it is, and the diameter of this grid is about 3 millimeters. Okay, just to give you an idea of the scale of the sample. And so this will be the final sample that is going to go in a transmission electron microscope. And this is an example of what you might be able to see if you do the sample preparation properly. So here's a TEM image of a cell, so yeast cell. And here you can see the cell wall, you can see the Golgi, you can see the nucleus, mitochondria, individual ribosomes, and so on. Okay, so yeah, so again a very powerful technique, but for a different purpose. Now, we already saw the concept of labeling proteins of our interest in fluorescence microscopy. What we can use a similar concept in electron microscopy, and in that we use something called immunogold staining, okay, or immunogold electron microscopy, which has the same idea. We have an antibody against a protein of our interest, right? Sorry. an antibody is going to bind to our protein. But on the other end of the antibody, instead of having a fluorophore, we have a gold nanoparticle. Okay, the gold is, as we discussed, it's electron dense, so it is going to scatter electrons. Okay, so this will appear as a black dot in our image. Okay, and here is just an example of the resolution we can get with immunogold staining. So here there are different components of the spindle pole body. Okay, these are four different proteins essentially of the spindle pole body, and we can label them separately with these antibodies and use them, and thus we can localize each of these proteins in the spindle pole. Right? You can see all these black dots are individual antibodies that have gone and bound. Similar thing here, but this is in scanning electron microscopy. So here is a bacterium, and they've used 10 nanometer colloidal gold for a surface adhesion protein. Okay. In scan electron microscopy, gold labels will appear as a white dot. Okay. Transmission electron microscopy, they're appearing as black dots. Here they'll appear as a white dot. Okay, some questions related to electron microscopy. What in biological samples diffract electron rays and create noise? Water is removed by treating the sample with a graded series of ethanol. Okay, this is called the dehydration step. This is the fixing. These are both fixing steps. This is the only dehydration step here. In TEM, the electron beam is focused on the specimen with the help of dash. Okay, the electron beam. So this is basically analogous to the illumination in a light microscope. So that it would be focused on the specimen with the help of the condenser lens. The coating of specimen with gold palladium is performed to dash. Okay, so it is essentially used to make the sample more conductive. This is the sputtering step in scanning electron microscopy. Okay, it's used to make the sample more conductive. It's used to increase contrast in scanning electron microscopy. What is the primary advantage of using an electron microscope over a light microscope? So the primary advantage would be high magnification and resolution. Which microscope technique is used to observe the internal structure of cells and tissues? So we want to observe the internal structure, we will have to use transmission electron microscopy, because scanning electron microscopy is only going to observe the surface. Which microscopy technique is used to observe the surface structure in high resolution? This would be scanning electron microscopy. Okay, so now that we have discussed the process of microscopy, there is just one more technique that was discussed in this week's content. Okay, that is called flow cytometry. Again, this is flow cytometry is a technique which allows us to observe different properties of a cell. Okay, such as things. So we would observe several properties of a single cell simultaneously, and yeah. So the main difference is here is that we can individually measure the property of a single cell, and that is done by essentially through this method called hydrodynamic focusing, where our cells are forced into the single cell suspension that individually pass through our detector. So here we have something called sheath fluid, okay, which is coming through the nozzle, and a sample, which is going to be cells in suspension. So we need a suspension of individual cells, and when this is passed through the flow cytometer, this will force it into a single cell suspension where they will go in front of the light source one at a time. And we can use this to detect properties of cell like fluorescence and scattering. So the single cell suspension is created through something called a fluidic system, which is transports particles of the fluid stream to the laser beam. Okay. Optimally, we want to look at the particles one by one. So this particle should be positioned in the center, and only one cell or particle should move through the laser beam at any given time. So how can we accomplish this? To do this, the sample is injected into a stream of sheath fluid. Okay, this is called the flow cell. Okay, this region is called the flow cell, and the flow chamber has a nozzle tip. And because of the nature of the flow, essentially what is going to happen is we're going to have a central column with our cells passing through it, surrounded by the sheath fluid, which is going to force it into a laminar flow. Okay. The flow of sheath fluid accelerates the particles and restricts them to the center of the sample core. This process is called hydrodynamic focusing. So here you can see we have sheath fluid and we have the sample. And if the sample is passed at low pressure, we can see that it forms this narrow strip, and we get these individual cells which are going as a single cell suspension. And as we increase the sample pressure, basically the width of the sample core increases, and we are less likely to get single cells, isolated single cells in our detector region. Okay. So hydrodynamic focusing basically requires a difference in pressure between the sheath fluid and the sample fluid. If we have low differential pressure, then it will be focused well. If we have high differential pressure, then we will lose the sample focusing. So as we saw, increasing the sample pressure increases the flow rate, right? By allowing more cells to pass at any given time. So some cells could pass through the cell laser beam off center and intercept the laser beam at a less optimal angle. But obviously, if we increase the sample pressure, the flow rate will be faster, and this will decrease the time required for the experiment. So we have to choose a flow rate depending on our application. So higher flow rate is generally used for qualitative measurements, whereas a lower flow rate can be used for more sensitive measurements such as things like DNA analysis. So we can do some questions. What properties of cell or particle can be measured by flow cytometer? We can look at things like granularity. Okay, we can look at things like granularity. We can use fluorescent markers to label certain surface properties. Okay, we can look at cell cycle analysis. Okay, we can look at the size of the particle. So there are a lot of different properties that we can measure using a flow cytometer. What light sources use in most flow cytometers? Usually we will use a laser. This is most commonly an argon ion laser. What are the three main systems in the flow cytometer? So we have the flow cell, okay, or the fluidics component. The second will be the optics component, which is the detector in the laser. And third is the electronics component, which we will discuss in the coming slides. What type of biological sample is best suited for flow cytometric analysis? Obviously, we need a single cell suspension, right, for flow cytometric analysis. So it is important that a sample is present as that single cell suspension. And what is the name given to the portion of the fluid stream where the cells are located? So that is the sample. It's basically the sample core, where the cells are located. So as we mentioned, we can look at multiple properties of the cells using a flow cytometer. So here we have a sample coming, encountering the laser. So here you can see we can detect various scattering from the cells. We can look at something called forward scatter. We can look at something called side scatter. We can also have various fluorescence detectors. So these are various fluorescence channels separated by dichroic mirrors. Okay, as we discussed in the fluorescence microscopy section, you can use dichroic mirrors to separate the emitted light into various fluorescence channels. So we can look at multiple fluorophores at the same time, which means you can label multiple properties of the cell and observe them. But first, we'll discuss this property called scattering. Now, scattering will tell us two things, okay, because we're measuring two different kinds of scattering. One is called forward scatter, okay, which is basically telling us the amount of light which is coming around the cell, and this is going to tell us the size of the cell. Okay. Forward scattering is going to tell us the size of the cell, whereas side scattering means light which is going to encounter organelles within the cell and be scattered from the cell, from within the cell. So what side scatter tells us is a property of the cell called granularity. Obviously, if the cell is more granular, then there are more objects within the cell to scatter light, right? And so we will have more side scatter. And forward scatter is obviously related to the size. If we have a smaller cell versus a larger cell, we will get more forward scatter if the size of the cell is larger. Okay. What now we can now do is basically plot a 2D image. Okay. We can have forward scatter like this. We can have side scatter on the y-axis. And we can get this 2D plot to separate types of cells based on the size and granularity. Okay, so if we were to run a blood sample, this is a very classical example, then obviously if we have granulocyte cells, these will be large and they will also be more granular. So you can imagine that these will have both high forward scatter as well as high side scatter. So if we have side scatter on y-axis, forward scatter on x-axis, then the granulocytes are high for both of these, so they will be present in the upper right portion of this graph. So this population is corresponding to granulocytes. Then we have monocytes, which are large cells but not granular. Right? So they will have high forward scatter but low side scatter. So this population will be monocytes. Small lymphocytes and lymphoblasts are not granular cells. They will have low side scatter. But because they're smaller, they will also have less forward scatter. So this population will be lymphocytes. And here we will have another population which is not really corresponding to forward scattering to a cell. And this will probably be cell debris. Okay. So thus we can separate cells into different populations just based on forward and side scattering. So in this process, we have not labeled any property of the cell. Right? We have not used any fluorophores, we have not done any labeling. We just directly pass the cells through a flow cytometer and we can get an idea of the relative populations of these different cells. Okay. So when does light scattering occur? Light scattering will occur when the cell interacts with the incident beam of light and is scattered both by the cell itself as well as by granules within the cell. Which key cell components compare? These are intercellular organelles. Light scattered in the same direction as laser beam is called forward scatter, or FSC. FSC is proportional to the size of the cell. Light scattered at 90 degrees to laser beam is called side scattering or SSC. SSC is proportional to the granularity of the cell. Okay. And obviously if we measure both FSC and SSC, or both forward and side scattering, we can differentiate different cell types within heterogeneous cell populations we just saw with blood. But as we discussed earlier, we can also look at different fluorescent markers within the cell. And how that is done is we have a cell which is either we have used an antibody as a fluorescent marker, or it is expressing some fluorescent protein, or it is fluorescing in some way. And as it fluoresces, it's going to encounter a laser. Okay? And as it passes through our laser, it is going to cause a pulse. Right? This is going to be proportional to the intensity of fluorescence which is present within the cell. Okay? So as a cell passes through the laser, we will get this profile of fluorescence, okay, with some height which is corresponding to the fluorescence intensity, height and pulse area. The height will correspond to fluorescence intensity, and pulse area will correspond to the size of the cell. Okay. Sorry. And if we look at the data that we can get from just, if we've just labeled one fluorophore, okay, then as the fluorescence increases, we will have a higher fluorescence signal. Okay? So on the x-axis, we have basically fluorescence signal. Okay? So the higher the amount of fluorescence, the further this will come on the x-axis. Whereas y-axis is something called events. And events is basically number of cells detected. Okay? So here you can see that if I have a cell, okay, two cells, one, and I'm looking at the expression of a fluorescent protein. And one cell has a very low expression, the other cell has high expression. Then for the cell with low expression, I'll get a peak here, whereas cell with high expression, I'll get a peak here. Okay, and the height of these two peaks would be related to how many of these cells are present within the population. So if the low expressing cells are more, then this peak will be bigger. But if the high expressing cells are more, then this peak will be bigger. Okay. So this is a way of representing flow cytometry data, which is called a histogram. Yeah, so here you can see events at low fluorescence intensity. So this is fluorescence intensity, this is events. So you can see most cells within the sample have low fluorescence intensity. On the other hand, if most cells have high fluorescence intensity, you will instead get a peak on this side, right? Because they have a higher fluorescence intensity. And so you'll see this peak shift. And so if we want to look at two different populations of cells, say I have stained and unstained, or my cell and a negative control, then I should be able to see a shift in the intensity. For example, here if I'm looking at the negative control and a positive sample, the black is my negative control. So because of my negative control, it should not have my protein of interest. And so the fluorescence intensity should be very low. Right? So all my events should come here at a low intensity. Whereas in my positive sample, okay, since this my protein of interest is present, most of my cells should express my protein of interest. So the peak should come at a higher fluorescence intensity, right? Because my protein is present in these samples. One thing to keep in mind if you're going to use multiple fluorophores is that we should try to avoid an overlap between the emission spectra of our fluorophores. Okay, if we're going to want to label two different properties of the cells, I want to label some surface protein, I want to label some internal protein, I will need to use two different fluorophores for this. But when I choose these two fluorophores, we should choose fluorophores that do not overlap in their emission spectra. Okay, For example, here you've used FITC and PE, and when we use these two, there is a slight overlap in these emission spectra, okay? And this can lead to false positives. So when we're looking at populations that have, if you want to look at populations that are both PE positive and FITC positive, this can lead to false positives, because essentially we are not measuring pure PE emission, we're also measuring some component of the FITC emission. So ideally we want to avoid the spectral overlap, right? We would want to choose fluorophores like this without overlap. Also, for flow cytometry, it's very important to use the correct controls so that we can analyze our data properly. So some recommended controls that we should use are unstained cells. Okay, so this is going to help us define things like cell size and cell granularity. This is just giving us a baseline reading for that. We can use live dead markers to isolate healthy cells from dead cells. Okay, we're making sure that the properties we are measuring are from the healthy cells. Single stain positive control. So if we want to look at multiple properties, or like use multiple fluorophores, and for each fluorophore we should have a single stained control. Okay, population cells which is stained only with that. Okay, so this will help us get an idea of the values, fluorescence values that we should be expecting for that fluorophore. And if we are going to use multiple fluorophores, then another control we should use is fluorescence -1 staining. Okay, which basically means we have these 4 fluorophores. Then I should also have for this a control in which this fluorophore is absent. Okay, but the other 3 are present. So this will help me define which is the positive population for this and which is the negative population. Okay, it's basically negative control for this. Okay, so these will all help us analyze the flow cytometry data better. So there's some questions based on this section of flow cytometry. When fluorescent compounds absorb a light energy and release excess energy, they emit photons. The characteristic wavelengths at which fluorescent compounds can be excited are called the absorption spectra. This is either absorption or excitation spectra. Longer wavelengths of light emitted by fluorochrome make up its emission spectrum. Which lasers commonly used? This is argon ion lasers. FITC and PE fluorochromes are excited by this emission wavelength. This is usually 488 nanometers. Fluorescent dyes, you can use multiple dyes. We usually use FITC, Psi3, Psi5, lots of dyes. Fluorochrome labeled antibodies are used to detect a protein of our interest. Okay. When cells labeled with fluorescent molecules pass through focused laser beam, what 2 types of light signals are generated? So this would be we are going to generate both the fluorescent signal as well as a scattering signal. Light emitted from a particle is collected by the detector, which is usually a photomultiplier tube or PMT. Electronic signal produced by detectors is proportional to the fluorescence of the cell. All flow cytometric measurements can be made simultaneously on a single cell. That's true. Particles must be in single cell suspension before flow cytometric analysis. It's also true, right? Because each measurement should come from a single cell. Now that we have collected our data from flow cytometry, There are many different ways in which we can visualize this data. Okay, and one concept that we should understand if we want to see a subset of our data is something called a gate. Okay? Gate is a numerical or graphical boundary that can be used to define the characteristics of particles to include for further analysis. So suppose this is the full population of cells that were present in our sample, but we're only interested in this population. Okay, so through FSC and SSC I'm able to separate the different kinds of cells present in my sample, but I am specifically interested in what these cells are doing. Then I can define a boundary, okay, within the software itself, which tells me that I only want to look at these cells. Okay, and so we would get fluorescence properties only of these cells. So you can imagine I have taken a blood sample, I have stained it with some fluorescence marker. My first step will be to run this FSC SSC plot. Right? I'm just going to run it through the scattering plot. Within the scattering plot, I am going to now set a gate. Okay? This gate is going to give a boundary on the FSC and SSC properties of the cells that I want to analyze. So now when I collect my data, okay, it is only going to come from samples which are within this gate. Okay? So this is telling me that I don't have to separately purify the lymphocytes from my whole blood sample. Instead, within flow cytometry, if its scattering properties are matching the gate which I have given, then it will be included in my analysis. So this can be useful for analyzing subsets of your sample. So yeah, a gate can be used to restrict the analysis to a specific population within the sample, right? Which is the definition of a gate. So as we discussed, there are many different ways in which to represent fluorescence data. So the most common would be a dot plot. So each of these dots is corresponding to one specific event or one specific cell. And since we're looking at two parameters, we have one on the x-axis, one on the x-axis, one on the y-axis. And so what we have is each plot, each dot will have a certain value of forward scatter, a certain value of side scatter, and that will define its xy position in the graph. Okay. So this is a dot plot with a gate is encompassing the lymphocyte population, as we just discussed. Another method of representation, if we're only looking at one property of the cell, okay, then we can use a histogram. So in the histogram, we basically have intensity on the x-axis and counts or number of events on the y-axis. Okay, and so these different peaks are basically corresponding to different populations within a sample. And within a histogram, we can also set gates, but these gates will look slightly different. Okay, so this is called a histogram marker. These M1 and M2 are called histogram markers, and these work in the same way as a gate, so they are defining the population. So I'm defining that M1 population is the population which has this much fluorescence intensity, and this is the M2 population. Okay, so these are called markers in histogram, which are basically separating these two populations. Now if I define these two markers, then I can also get statistics for these two populations. So here I can see in the M1 marker, I'm getting 619 events, right, over here. Percent gated is 21.41, it means 21% of the total population is present in the M1 gate. Right? And here it is giving me what is the fluorescence characteristics of it. Okay, so here it is telling me that the mean fluorescence intensity, okay, for this population is 3.75, whereas in the M2 gate, okay, which is this second population, I have 2,200 events, okay, which is 78% of the total, and the mean value of fluorescence for this is 224. So you can see there's a very obvious difference between the M1 and M2 populations. And since here we are looking at a surface marker called CD3, so CD3, I am having some antibody which is against CD3, and the CD3 antibody is conjugated to a fluorophore called FITC. And so since I'm looking at this one property, I can see that this population which has low fluorescence intensity is going to be CD3 negative, whereas this population is going to be CD3 positive. So if we look at these figures, we can determine which percentage of the lymphocytes is not CD3 positive, right? So this is the CD3 positive. So this population is not CD3 positive. Okay. So the percentage is the percentage within the M1 gate, which is 21.4. Okay, and then it's asking which percentage of total events is CD3 positive. So that is what percentage of the events is present in the M2 gate. And that is 78.59. Yeah, so this is the kind of statistics we can do by introducing these gates in our analysis. Again, as we discussed, if we have two parameters that we're looking at, we can make a dot plot. And in our dot plot, since this is one parameter and this is one parameter, we can divide our dot plot into 4 quadrants. Okay. So if this is parameter 1, this is parameter 2, in this quadrant, 1 and 2 are both low. Okay. In this quadrant, 1 is high and 2 is low, right? This is high amount of this parameter 1. So for example, this is high CD3, but it is low for this other marker, which is CD19. In this quadrant, however, it has a high amount of CD19. That means 2 is high, but the amount of CD3 is low. Okay, so these are both positive for one marker, but they're not positive for both markers. So these are the single positive populations. In this quadrant, in the top right corner, right, both parameters are high. Okay, so this is called the double positive quadrant. This is called the double negative quadrant, and these are the two single positive quadrants. So in this experiment, you can see they're looking at one surface marker CD3. This is using an antibody conjugated to FITC. This is CD19, which is another surface marker. This is conjugated to PE. So they're looking at the expression of two different fluorophores, and they have gotten some double negative population, some single positive populations. This is CD3 positive, this is PE positive, and there are few events which are double positive. Same thing, once we have defined these 4 quadrants, we can get quadrant statistics the same way we got histogram statistics. So here we get upper left. Okay, this is upper left. So this is positive for CD19. Okay. This has 10% of the population. Upper right, this is the double positive. Right? This is the double positive population. You can see that there are very few events, and same thing here, it's saying it's 0.1% of the total. This is lower left, which is the double negative quadrant, and this is the lower right, which is the CD3 positive quadrant. You can see most of the cells are present in the CD3 positive. They only have CD3, they don't have CD19. So here they're asking what percentage of lymphocytes are CD3 plus, CD19 minus. Right? So here we are going to look for lymphocytes which have this marker but don't have this marker, which will be basically the quadrant which has high CD3 and low CD19, which is this quadrant, at the lower right quadrant. And we can see from the quadrant statistics So this is 79.57% of the lymphocytes. We don't have to make quadrants. We can just make gates like this with 4 regions. Okay? It depends on the nature of the distribution of your events. Okay? And same thing, if we make regions, then we can get region statistics. Okay? Which will basically, in the same way as the quadrant, it will give us an idea of what percentage of the population is there in each region. Okay. Few more questions. What does the horizontal axis in a histogram represent? It represents intensity. Okay. A dot plot can be used to display 2 parameters, right? So you have one on the x-axis and one on the y-axis. The population in the lower left quadrant is double positive population. Okay. Just remember, a dot plot, the first parameter will increase like this, second parameter will increase like this. This is the lower left quadrant. So this is actually the double negative population. Okay, it is not the double positive population. Okay. Same deal, is the double positive population false. And just quickly we'll discuss probably the most most commonly discussed flow cytometry experiment, which is the apoptosis assay. Okay. The apoptosis assay uses two it's usually called the annexin V or annexin 5 propydium iodide assay. Okay. So annexin V is an anticoagulant protein which binds to phosphatidylserine. Okay. It is usually conjugated with a fluorochrome, so you will usually see it referred to as annexin V. Let's see. Okay, annexin V itself is not a fluorophore. It has to be conjugated to a fluorophore, so it will usually be conjugated to a green fluorophore, like FITC. Now, what annexin V binds to is something called phosphatidylserine, which is a phospholipid within the cell which is within the cell membrane. But for our cell membrane is an asymmetric cell membrane, which means the distribution of lipids on both the inner and outer leaflets is not the same. So in a healthy cell membrane, phosphatidylserine is present only on the inner leaflet of the cell membrane. It's not present on the surface. So if we incubate our cells with annexin V, in a healthy cell, there will not be any annexin V staining because there's no phosphatidylserine present on the surface. However, during the process of apoptosis, the asymmetry of the bilayer is disrupted. Okay? And phosphatidylserine will become exposed to the outer surface. Okay, and if this is the case, then annexin V can come and bind to this. So in early apoptosis, we will see this flipping of phosphatidylserine, and annexin V can come and bind to it. And as the apoptosis process progresses, the membrane starts to disintegrate. Okay, so we start getting these pores in the membrane. And in this case, what this means is that if we use another dye called propydium iodide, okay, propydium iodide actually binds to DNA. Okay, when it binds to DNA, it gives an orange fluorescence, orange or red fluorescence. But this cannot pass through an intact cell membrane. So only when the cell membrane starts to disintegrate, propydium iodide will be able to enter the cell and bind to the DNA. Okay. So by using these two markers, we can determine the number of apoptotic or necrotic cells within a population. So this is what PI and XNV data might look like. An XNV is on the x-axis, PI is on the y-axis. In a live cell, right? In a live cell, Phosphatidylserine is not exposed. There are no pores in the membrane. So neither of these dyes will be able to bind, right? So live cells will be in the double negative quadrant. This will be the lower left quadrant, double negative, which contains live cells. Um, in early apoptosis, remember, phosphatidylserine is flipped to the outer leaflet. So an X and V will be able to bind, but there are no pores in the membrane. So PI will not, right? PI will not be able to enter the cell. So in early apoptosis, our cells will be positive for an X and V, but negative for PI. Right? So this will be the lower right quadrant. Okay, the lower right quadrant will have early apoptotic cells. This is an X and V positive, PI negative. Ideally, we should not get cells which are positive for propydium iodide but negative for an X and V. Okay, so this is basically corresponding to naked nuclei. So these are cells without a membrane. Okay. So this population should be very, very minimal. But in late apoptosis, as we discussed, uh, obviously the flipping of phosphatidylserine has already taken place. But now there are also pores in the membrane which allow propydium iodide to bind to the DNA. So this will be the double positive population. Okay, late apoptotic or necrotic cells will be the double positive population, which is present in the upper right quadrant. Okay, so this will be the quadrant corresponding to late apoptotic or necrotic cells. And now you can use this to compare different kinds of drugs and so on, and actually quantify the amount of cell death that we get as a result of our treatment. So far we've discussed flow cytometry, but there are some machines which can go one step further and actually sort the cells. So this is something called FACS, fluorescence assisted cell sorting. Okay, and the initial idea is the same, and we have a nozzle tip through which a stream of cells is going to pass. Okay, and then we have a break off point. And what is actually going to happen is, depending on the population of cells that we detect here, so we have red cells and green cells. What is going to happen is that by virtue, it is going to basically impart a charge to these droplets, okay, and then deflect them based on the population into two different collection tubes. Okay. So it is going to sort the cells depending on the signal that it has detected over here. So this is a very powerful technique for sorting of live cells. And just quickly, we will do some questions for this. In flow cytometry, the side saccada channel captures data related to the cell granularity, as we discussed. Cell shape, size, etc. is captured by the forward scatter. Selection of data acquisition from a distinct fraction of cells and from cell population can be achieved by gating. Okay, this is the definition of gating. In a cell cycle analysis by flow cytometry, cells are stained with DNA binding dye. Okay, so this is usually done by staining the cells with propydium iodide. Okay, and we can then follow the cell cycle of different cells and look at what population of cells are present in different stages of the cell cycle. Because the main difference in the cell cycle is the amount of DNA which is present within the cell. In apoptotic cells, an X and V binds to phosphatidylserine as PS in apoptotic cells translocate to the extracellular cell of the plasma membrane. Right? That's what we discussed. Compensation is required in flow cytometry because of spectral overlap between two fluorophores. Okay, remember we discussed we should not have spectral overlap, but if we do have spectral overlap, then we have to basically compensate for that. Okay, in our data analysis. Yeah, that was as much as I had to discuss today. As always, if you have further questions midweek, you can always use the discussion forum, or you can always join these problem solving sessions to get any doubts that you have cleared. Okay, thank you and good night.